Essentials in Biotechnology. Learning objectives. 1. Understand the fundamental principles of DNA recombination and the role of restriction enzymes in isolating specific DNA segments. 2. Comprehend the significance of plasmids as vectors for transferring genes of interest into host cells for amplification. 3. Understand the process of colony selection and how marker genes are used to identify transformed cells in recombinant DNA technology. In the previous videos, we explored the fascinating double helical structure of DNA and how its strands are oriented in an antiparallel manner. Additionally, we delved into the cell's central dogma, which explains the process of protein production. This understanding underscores the importance of having access to a DNA structure that corresponds precisely to a specific protein sequence. When working with DNA, one of the main challenges is isolating a specific segment from the rest of the DNA sequence. Initially, there was no guarantee that nucleases could cut DNA in a way that specifically excluded regions inside the genes. A groundbreaking milestone in the field was achieved when scientists isolated a specific type of enzymes from bacteria that could cleave DNA at precise sequences. This type of enzyme was named restriction enzymes, and they recognize specific sequences in double-stranded DNA known as restriction sites. Let's focus on the EcoR1 restriction site, a specific recognition sequence for one of well-known restriction enzymes. Upon examination of the upper strand, you will observe the sequence GAATTC, from 5' to 3', and on the lower strand, the reverse complement of GAATTC, which is in the opposite 5' to 3' direction. There are various other enzymes, each with its own specific recognition site. These enzymes, like EcoR1, recognize and cleave DNA at precise sequences, facilitating the process of genetic manipulation and DNA recombination. Now let's consider a DNA molecule containing the recognition site for EcoR1. Suppose that there is a site with the sequence GAATTC. Notably, on the top strand, from left to right, 5' to 3', GAATTC is observed, and on the bottom strand, from right to left, also 5' to 3', GAATTC is present. When the EcoR1 enzyme encounters this specific sequence, it recognizes this site and initiates the process of cleaving the DNA at the restriction site. This cleaving results in two different DNA segments, often referred to as DNA fragments. These fragments have unpaired ends, known as sticky ends. They can easily bind with complementary sequences in other DNA molecules. This unique property of the sticky ends allows the fragments to join with other DNA segments, facilitating DNA recombination and genetic engineering. Let's consider a scenario where a specific gene is located within a DNA molecule, and two restriction sites flank the gene on both sides. When the restriction enzyme encounters the DNA segment containing the gene, it recognizes and cleaves at the specific recognition sites on either side of the gene, separating the gene segment from the rest of the DNA molecule and generating smaller DNA fragments. This process of cleaving the DNA at the restriction sites allows researchers to isolate and extract the gene of interest from the larger DNA molecule. Now the isolated gene can be transferred to another cell to be manipulated. To transfer the gene of interest into another cell, a vector is required. In the context of bacterial cells, plasmids are commonly used as vectors. Plasmids are small circular DNA molecules that exist naturally in bacteria and can replicate independently from the chromosomal DNA. For this purpose, the vector should contain a specific sequence called the origin of replication, ORC, which initiates the replication process. The ORC ensures that the plasmid can replicate within the bacterial cell, allowing for the maintenance of the gene of interest as the bacterial cell divides. Additionally, the plasmid should contain a marker gene, serving as a selectable marker to identify bacterial cells that have taken up the plasmid. Common marker genes include those conferring antibiotic resistance. In cases where the plasmid contains a restriction site for the same enzyme used to isolate the gene of interest, the plasmid can be cut by the same enzyme. When the restriction enzyme cuts the plasmid at its specific recognition site, it cleaves the circular DNA molecule, resulting in a linear DNA fragment. The sticky ends created by the restriction enzyme are essential for the subsequent step of gene insertion. Now we end up with two DNA fragments, each with sticky ends on each side. When the gene of interest and the linearized plasmid with sticky ends are present in the same tube or reaction mixture, they can bind to each other. This process, which is facilitated by the complementary sequences of the sticky ends, can be observed closely. The complementary sticky ends of both DNA fragments are capable of base pairing. By adding the enzyme DNA ligase, two fragments can be joined together in a process known as ligation, leading to the creation of a novel DNA molecule called recombinant DNA. This recombinant DNA contains genetic material from two different sources. Now it is time to introduce the recombinant plasmid into suitable host cells for their amplification. To perform the transformation, the recombinant DNA must be introduced into the cell culture. Next, the cell culture is allowed to incubate, enabling the plasmid to enter the cells. Then, the cells undergo a sudden environmental change, often through heat shock or electroporation, to facilitate plasmid uptake. Finally, the cells are cultured under specific conditions that promote the multiplication of the transformed cells. One of the crucial stages in recombinant cell production is colony selection. This process involves a method to distinguish cells that have successfully acquired the plasmid from those that haven't. The marker gene plays a significant role in this step, as it allows for the identification and isolation of transformed cells. In a cell medium without any antibiotics, all bacteria can survive, irrespective of whether they have the plasmid or not. However, in a medium containing a specific antibiotic, only the bacteria that have taken up the plasmid with the antibiotic resistance gene can survive. This selective survival enables the identification and isolation of the transformed cells for further analysis and use. After the selection stage, the correct colonies need to be transferred to another growth medium to facilitate their proliferation. Next, the expression of the gene of interest should be induced by adding a specific substance. This induction leads to the transcription of the gene and translation of the corresponding mRNAs. Now the cells containing the exogenous protein can be collected from the cell culture. By lysing the cells, their contents become accessible. Finally, the proteins can be separated from other cellular components using various methodologies. This separation allows for the purification of the desired proteins for further study and application. Through this presentation, we have explored the key principles of recombinant DNA technology, encompassing DNA recombination, plasmids as vectors, and colony selection. By understanding these fundamental concepts, you are equipped with the knowledge to manipulate genetic material and create novel recombinant DNA molecules. After watching this presentation, you should have a solid foundation in biotechnology, which will enable you to apply these principles in various research and practical settings.